we are going to see about an important topic in plant physiology, that is with regard to the phytochromes. And phytochromes, as you know, is involved in light reception, and based on the changes what is occurring in phytochromes, lot of physiological functions are being activated in plants. And based on this, the morphogenesis of plants is being divided into broad two classes, that is scotomorphogenesis and photomorphogenesis. Whatever morphogenesis occurs under the influence of light, we refer to it as photomorphogenesis, and where no light influence is there, that can be classified under scotomorphogenesis. So wherever photomorphogenesis is involved, that means light is involved, lot of morphogenesis processes are occurring, and that is being under the influence of certain light receptors, and among these light receptors the prominent one is phytochrome. And phytochrome has been extensively studied, and if you check with the photochemical and biochemical properties of the phytochrome, you will know that it is a blue protein pigment with 125 kilodalton, and it was first discovered in 1959. But before the discovery of this protein in its structural form itself, they have had a clue on the function of phytochrome, especially the discoveries of 1930. There they have shown that red light can induce seed germination. That means that red light can influence a particular process in plants, that is a seed germination. And Lewis Flint in US, in USDA, that is United States Department of Agriculture, he only basically studied with the lettuce seed germination, and he found that the germination can be stimulated by red light, and the germination process can be inhibited by far red light. So that was one of his major finding. And Borthwick in 1952, he studied about more about the phytochromes. He found that it is a single pigment existing in two interconvertible forms, the red form and FR form. That is the form which can absorb red light and the form which can absorb far red light. So there is two forms of phytochrome. That was the finding of Borthwick in 1952. And this is the experiments which has been conducted by Lewis Flint. The lettuce seeds were germinated, and they found that in dark there was no germination, but when it was kept in red light there was a profuse germination, and when far red light was given immediately after red light, the germination process was not happening. So this was a clear evidence for that red light is promoting seed germination in the case of lettuce, but this effect is reversed by the application of far red light. And as you see here, phytochrome interconverts between red and far red forms. Here the red form is a blue pigment, and the PR form gets converted into PFR, that is green, and the conversion takes place by the absorption of red light, and the PFR form can come back into the blue form, that is the PR form, after absorption of the far red light. And dynamic equilibrium also exists between these two states, that is a total conversion is not taking place. The PR form is not totally getting converted into PFR form. There is an equilibrium existing between the two states, and we call it as a photostationary state. Partially one portion gets converted into PR form, simultaneously some portion gets converted into PFR form, and there is an equilibrium existing between these two states, which we call it as photostationary state. And in between the PR to PFR formation, it is told that we have many short-lived phytochrome intermediates in between these two PR and PFR forms. Whether intermediates play a role in initiating or amplifying phytochrome response under sunlight, which contains mixture of all visible wavelengths, that is not still unknown. mainly we have studied about these two states, but the states in between the conversion of PR to PFR form, whether that has got any influence on any physiological functions, that is still under study. And here you can see what is the distinguishing features between PR and PFR form. The PR form absorbs light only in the red region with a wavelength of 666, and PFR form can only absorb light in the far red region of 730. So these are the absorption spectra of both red and far red form. PFR is the physiologically active form. If you compare between these two, PR and PFR form, the PFR form, after absorbing the red light, which PFR is formed, that is the physiologically active form. The ratio between PFR and PR determines the magnitude of response. Also, it has been found that not only as I told you, there may be the intermediates may be playing a role, or even the ratio between PFR and PR also has a can determine the magnitude of the response. So it is not that PFR is active and it is solely responsible for the physiological functions, but it may be the ratio between the PR and PFR forms. And more studies were conducted in Arabidopsis mutans, and from the mutants it was found that the phytochrome is a dimer composing of two polypeptides. They had that the detailed structure of this will be studying in the coming slides, and basically you know that it is a dimer composed of two polypeptides, and studies were conducted by William Ziegelman, again in USDA, that is United States Department of Agriculture in USA, and he studied in young etiolated seedlings of cereals. He found that was one of the richest source of phytochromes, that is the young etiolated seedlings of cereals, which are rich source of phytochromes, and the total extractable protein in the form of phytochromes available was 0.2%. If you try to take out the total protein from the plant, of which only 0.2% would be the phytochromes. And when as already we told, it is a dimer. It consists of two subunits, one chromophoric portion and apoprotein portion. Chromophoric portion is basically responsible for light absorbing, and apoprotein portion is the polypeptide chain, and this together forms the complete phytochrome. And if you try to see about the synthesis of phytochrome, you can see that the precursor from which phytochrome is synthesized is aminolevulinic acid, 5-aminolevulinic acid, and this is a common precursor for both chlorophyll and phytochrome, and it is synthesized from this precursor. The assembly of apoprotein with its chromophore is autocatalytic. I already explained to you earlier in the earlier slide. It consists of two portions, that is the chromophoric portion as well as the apoproteinaceous portion. But once these are synthesized separately, they combine together autocatalytically. They come together and they combine together to form the complete protein. The PC apoprotein is synthesized, the phytochrome apoprotein is synthesized by expressing in Saccharomyces yeast cells. So, the researchers have succeeded in expressing the gene responsible for the synthesis of phytochrome apoprotein in Saccharomyces, that is the yeast cells, and synthesizing the apoprotein separately. So, this gives you a better picture of the structure of chromophore. You can see that this is the detailed structure of the phytochrome, and here we have a chromophoric portion. This is the long chromophoric portion, and this is the apoproteinaceous portion, and the chromophore is attached to that. And basically, when light is absorbed, or when the red light is absorbed, there is a conversion. Upon absorption of light, the PR chromophore undergoes cis trans isomerization. So what you see over here in the upper portion is the cis isomer, and when light is absorbed, it can get converted into trans isomer. So on absorption of red light, it gets converted into PFR form. So major change taking place is in this area, that is between carbon 15 and carbon 16, and the conversion of red form to PR form to PFR takes place. And if you say that, if you ask me whether only change is taking place in the chromophoric portion, it is not. Even change can take place in the apoprotein portion also. Apoprotein also undergoes changes in far red from the difference in their susceptibilities to proteases and in phosphorylation of exogenous protein kinases. So, basic change taking place in the apoproteinaceous portion is that the susceptibilities to proteases changes, and also in phosphorylation of exogenous protein kinases. So, these are the major change taking place in the apoprotein portion. The change taking place in the chromophoric portion is between carbon 15 and carbon 16, and based on that change the cis isomer gets converted into trans isomer on absorption of the red light. This is a little more detailed structure of the phytochrome, and this is the holoprotein of phytochrome, the complete protein of phytochrome, showing various functional domains. The chromophore binding site and PES sequence are located in the N terminal. So, this is the N terminal, and the chromophore binding site, this is the chromophore binding site, and the PES sequence, you can see this is the PEST sequence, that photodegradation area. This is the PEST, this is given in this purple color. PES sequence are located in the N terminal. This is the N terminal, this is the C terminal. So, this confers photosensory specificity to the molecule, that is whether it responds to continuous red or far red light. So, the absorption of red and far red light occurs in this N terminal and based on the chromophore, which is located on the N terminal. Now, coming to the C terminal domain, it contains the dimerization site. So, this is the dimerization site. I told you this is a, if you see the structurally, this is a dimer, and the dimerization occurs over here. Also, you can see a ubiquitination site, and also you can see a regulatory region. So, these are the three specialties of the C terminal. We have the dimerization site, we have the regulatory region, the phytochrome is regulated at this portion, and we have the ubiquitination site. So, this portion is basically responsible for signal transmission. So, signal transmission, after the change occurs in this region, based on the phytochrome AB specificity or on the absorption of the red and far red light, the signals are being transmitted from the C terminal. And if you see the size wise difference, the N terminal has got 74 kilodalton and the C terminal has got 55 kilodalton. So, this is the detailed structure of the phytochrome. And basically two types of phytochromes are identified, type 1 and 2. Type 1 is 9 times more abundant than type 2 in dark grown seedlings, and in light grown pea seedlings the amount of two types are equal. So, if it is in dark, type 1 is the dominant type as compared to type 2, but in light the both are of equal quantity, and the two types are distinct proteins. They have their own proteinaceous structure. These two are different as far as the polypeptide is concerned or the apoprotein is concerned. Now, how can we localize the phytochrome in tissues? Because it is being totally synthesized. What we know is that the phytochromes are synthesized almost in all parts of the plant. We can have a look on this portion. That is, if you try to examine in a germinating seedling, this is the variation of the phytochrome content what is observed in different areas. And the highest phytochrome content would be at the elongation zone, that is just below the first node. That is the highest phytochrome content what you see over here. And even the root tip also, root elongation area also, you can have a comparatively high phytochrome content. And in between you have a comparatively lower phytochrome content. So, if you try to examine a germinating seedling, almost all portions of the seedling has got the phytochrome content is there. So, synthesis takes place, and if you want to specifically localize the phytochrome content in the tissues of the seedlings, there are different mechanisms. One of the mechanism is that phytochrome can be detected in tissues spectrophotometrically. You can go for spectrophotometric detection, and from that studies it is found that phytochrome is high in meristematic tissues or regions which were recently meristematic, and that is why we are seeing it in highly in the tip portion as well as in the root tip portion, shoot tip portion and root tip portion. Another way of localizing phytochrome in tissues and cells is through antibodies. If you can develop the antibodies, that antibodies will specifically go and bind at a higher proportion to the phytochrome where it is localized at a high rate. So from that also you can localize or find out the regions where high content of phytochrome is there. And one of the other method which generally researchers follow is through the promoter genes. And if you can just attach the Gus gene along with the phytochrome genes, wherever the phytochromes are synthesized, the Gus genes also get overexpressed. And you may be knowing that when more of Gus gene expressing, more coloration would be occurring over there. One pink purple color will be developing, and based on the coloration you can specify or come to a conclusion where exactly more of phytochromes are synthesized. Now we will come to the ecological functions with regarding to the phytochrome. The involvement of phytochrome is mainly with regarding to some of the ecological function of shade adaptation. Phytochrome is encoded by a multi-gene family, and if you see, there are different genes are involved, phytochrome A through phytochrome E, that is phytochrome A, B, C, D, E. So 5 specific genes are involved. Despite the great similarity in the structures, each of these phytochromes performs distinct roles in the life of the plant. And phytochrome, the prominent phytochrome, we have already earlier we have seen that we have categorized phytochromes into 2 types, type 1 and type 2. And under type 1 we have specifically type A, phytochrome A, and under type 2 we have phytochrome B, C, D and E. So you should know that there are 2 types of phytochromes, type 1 and type 2. Under type 1 we have phytochrome A, and under type 2 we have phytochrome B, C, D and E. And if you examine these 2 types, that is the type 1 and 2, they are mutually antagonistic. They have different function, the antagonistic function, they promote antagonistic function, and how exactly it is being done is explained in the this particular figure. Here you are seeing a continuous elimination. So here it is a phytochrome B. When red light falls into that, see here, red light is also falling, far red light is also falling. This is without any shade. This plant is remaining in the open condition. But the same plant, phytochrome B, this is a what you say sun loving plant, and this sun loving plant is under a shade. Here the prominent phytochrome is B, that is the type 2 phytochrome. So the shade loving sun loving plant will have more phytochrome B, which is type 2, and a shade loving plant will have more of type 1 phytochrome, and that is phytochrome A. Now when phytochrome B containing sun loving plant is outside or in the sun exposed areas, the red light will be falling simultaneously, far red light also will be equally falling. So the conversion of PR to PFR and PFR to PR will be in equal level, because there is no hindrance for both red light and far red light, and the level of PR and PFR will be almost at equal level. But coming to the shade condition, you can see the amount of red light reaching the plant would be less. From the arrow itself you can find that whatever intensity is reaching the leaf, the canopy, will not be the same what it is coming down to the plant below the canopy. But the far red light can enter the canopy or penetrate the canopy and reach to the plant. This is what exactly happening over here also. So again try to remember that this is a shade loving plant, this is a sun loving plant. Shade loving plant will be having more of phytochrome A, and sun loving plant will be having more of phytochrome B. This is type 2 phytochrome, this is type 1 phytochrome. Now when more of far red light is reaching, the phytochrome PFR form will be converting into PR form. PFR form would be converting into PR form. Again, same situation here. More of far red light reaching over here means more of PFR will be converting into PR form. Now, why already in the previous slide we explained that these two phytochromes are having antagonistic function. I am trying to talk of that antagonistic function. Now, when more of the PR, here the PR would be more and the PR level would be more, because more of far red light means PR is getting converted into PFR is getting converted into PR, PFR is getting converted into PR. More of PR means the phytochrome over here is phytochrome A. It will not contribute towards the elongation of the seedling. It will not, I want to repeat again, it will not contribute towards the elongation of the seedling. But here, on reception of the far red light, the PFR form is getting converted into PR, but here instead of the phytochrome A, it is phytochrome B, and the phytochrome B will be contributing towards the elongation of the seedling, so that the seedling emerges at a faster pace and try to emerge out of the canopy and try to receive more of sunlight. And that way they have antagonistic function. Phytochrome have antagonistic function with respect to phytochrome B, because this does not contribute towards stem elongation, but phytochrome B contributes towards the stem elongation. So, this is an opposite function, one contributing towards the stem elongation and one contributing not towards the stem elongation. I can repeat this once again, because it may, if it finds it difficult to grasp. This is a condition wherein the plant is remaining outside the canopy. This is a sun-loving plant. These are two different types of plants. This is a shade-loving plant and this is a sun-loving plant. Both are remaining under the canopy. As it is remaining under the canopy, more of far red light will reach because of the higher wavelength. Shorter wavelength, the red light gets disrupted in between because of the canopy. Far red light without much disruption, it reaches to the plant. More far red light reaching means the conversion of PFR to PR takes place, and PR has in both these cases more PR will be accumulating instead of PFR. But here the phytochrome in the sun loving plant, it is phytochrome B, and in the case of shade loving plant, it is phytochrome A. Since it is phytochrome A, it will not contribute towards the elongation of the seedling out of the canopy, but in the case of sun plants, it will be contributing towards the elongation of the seedling out of the canopy, so that it can emerge out and receive more of sunlight. So, even though the phytochrome is a PR form in both these cases, since it is phytochrome A in shade plant and phytochrome B in sun loving plant, they show different actions. So, in this respect we call that they have an antagonistic function. And if you here in earlier slide we have seen regarding to phytochrome A and B, and if you ask me whether phytochrome C, D and E has any specific roles, they are much less studied. That is why it is being explained that like phytochrome B, phytochrome D also plays a role in regulating leaf petiole elongation as well as in flowering time. But comparatively less studies have come out with regarding to phytochrome C, D and E. The major studies have been conducted with regarding to type 1 and type 2, and out of type 1 it is only phytochrome A. And in type 2, although we have phytochrome B, C, D, E, the major studies has been conducted with regarding to phytochrome B. So, similar analysis support the idea that phytochrome E acts redundantly with the phytochrome B and phytochrome D in these processes, but also acts redundantly with the phytochrome A and phytochrome B in inhibition of internode elongation. So, some cases phytochrome like the example of phytochrome E, it may not be acting independently, but it may be acting redundantly either with phytochrome B or phytochrome A. Phytochrome C, D and E appear to play roles that are that are for the most part redundant with those of phytochrome A and B. So that means they don't have much of independent role, but they play roles redundantly with phytochrome A and B. So the major forms of phytochromes are phytochrome A and B, and the other forms C, D and E have roles which they play redundantly with that of A and B. This is another important ecological function controlled by the phytochrome, that is the circadian rhythm. That we will be explaining in the next session. This is also equally important because many of the plant functions are going in a rhythm which we refer to as circadian rhythm, and this is also controlled by phytochromes, and we will be explaining in the next session.